Today we're going to finish the week by going over pathogens and infections of the respiratory tract. We're going to cover the anatomy and defenses of the respiratory system, as well as the microbiome that is normal to the upper respiratory tract. We're going to cover the common cold, which is caused by many different types of viruses, and then cover specific pathogens and the diseases that they cause. So strep throat or streptococcal pharyngitis is caused by Streptococcus pyogenes. Whooping cough is caused by Bordetella pertussis. Influenza is caused by the influenza virus. And COVID-19, our newest respiratory system infection and pandemic, is caused by a novel strain of the coronavirus that has been called SARS-CoV-2. All right. So here we have regions of the respiratory tract, and I'll point out the places that are most commonly sites of infection. So if you have an upper respiratory tract infection, that means your nose or your oral cavity is inflamed or the back of your throat. The larynx is also part of the upper respiratory tract. So if you have laryngitis, then that would be an inflammation of the larynx. So that's more of your voice box, which is down on this part of the throat, whereas pharyngitis, a true sore throat, is at that very back of where the oral cavity meets the nasal cavity. In this location we call the nasopharynx. The lower respiratory tract includes everything from the trachea and below, and the lower respiratory tract should be sterile. The lower respiratory tract includes the trachea, which is also called the windpipe. The trachea leads to two bronchi, which are the branches that go into the lungs, and the bronchioles and the alveoli are the deepest parts of the lungs. The alveoli are going to be the air sacs where gas exchange occurs. And so if these fill up with fluids, then you can't get the diffusion of oxygen into your bloodstream, and that is how pneumonia can cause death. So having fluid fill up our lungs or fill up our alveoli is very dangerous. Now, oftentimes we'll have a sinus infection, which feels like an upper respiratory tract infection, but I want to point out that the sinuses are just hollow cavities in our cranium or in the skeleton structure of our skull. So we have these sinus cavities, and they are adjacent to the upper respiratory tract, but they're not a part of it. The sinuses can drain into the nasal cavity and down the back of the throat, but they are not a functional part of the respiratory system. All right. The respiratory tract has many natural defenses. There's a lot of mucus secretions that come from the nose and the nasal cell linings of the nasal cavity. So mucus secretions move mucus downwards toward the throat, so we can either cough it out or swallow it. Hair in the nasal cavity is also a physical barrier. We have cilia on the trachea, the cells that line the trachea. So the cells, the epithelial cells that line the trachea and the bronchi have cilia, and that helps move mucus in an upward direction. And so the cilia beat and the mucus moves up and out of the throat. We also have other first-line flushing mechanisms, including coughing, sneezing, and swallowing. There are macrophages, large white blood cells that are phagocytic, that circulate in the alveoli of the lungs, making sure that nothing gets in there, or if it does, it doesn't stay. And then we have antibodies or immunoglobulins in our mucus secretions as well. All right, so an antibody and a mucus secretion, that would be a third line of defense. Same thing with macrophages. And then all of these would be first line host defense mechanisms. There are microbes in the respiratory tract, but only in the upper respiratory tract. So the trachea, the bronchi, and the alveoli of the lungs should be sterile in a healthy individual. The first pathogen that we're going to go over is Streptococcus pyogenes. In labs, sometimes or in clinical labs, this might be called group A strep. Streptococcus is a genus of bacteria that are gram positive. So if we did a gram stain, we'd see purple cells under the microscope. The cells are arranged in a staphylococci arrangement, so that means you see chains of cocci or circular shaped cells. It is a facultative anaerobe that grows by sugar fermentation, so make sure you review what fermentation is and how that's different from aerobic respiration. It is a capsule former, and it's hemolytic. In lab, you learned that this is an example of a beta hemolytic, which means the hemolysins produced by this pathogen will lyse completely the red blood cells in its surroundings. So it produces a hemolysin called beta hemolysin. All right, as for the 3 major virulence factors you need to know, we've got capsules written down, but since it's not gram-negative, it will not have endotoxin, and since it's not bacillus or clostridium, it will also not form endospores. So Streptococcus pyogenes is the cause of streptococcal pharyngitis and also the #1 cause of bacterial pharyngitis, or an inflamed and infected throat, that's what this means, caused by a bacterium. There are many cases of pharyngitis that are caused by viruses, but if you have bacterial or streptococcal pharyngitis, you will see white spots on the tonsils. There will be a bacterial smell associated with the infection, and there's often a much higher fever and a very, very painful throat with difficult, painful swallowing. Pharyngitis is a very dangerous infection in that it can lead to other infections or disseminate to other parts of the body. If it spreads to the skin and you have a rash, that's called scarlet fever. And rheumatic fever is when the heart valves get damaged or the endocardium of the heart is infected. And so this would then reach the circulatory system and be likely to become septic. And so complications of strep infections are more dangerous than the initial sore throat. So this right here is that scarlet fever, and here is an infected heart valve, which would be the rheumatic fever. All right. Streptococcus pyogenes has a number of virulence factors. We've already mentioned the capsule, and we've gone over and a lot of times how capsules can contribute to pathogenesis. It also has a protein called M protein that you see right here that can aid in adhesion and also aid in evading phagocytosis. Lipoteichoic acid is a normal part of the gram-positive cell wall, that thick cell wall made of peptidoglycan that you see here. Lipoteichoic acid is that molecule that holds it together and anchors it to the cell membrane underneath. And if the lipoteichoic acid extends even outside the capsule, then it can further contribute to adhesion to host tissues. I've mentioned before that the organism has hemolysins. They're called streptolysin for Streptococcus hemolysins. And that beta hemolysin is the one that damages red blood cells. And Streptococcus pyogenes has a number of other tissue-degrading exoenzymes like streptokinase, DNase, and a variety of proteases or protein-degrading exoenzymes. It's got a lot of virulence factors, and strep throat can be transmitted through droplets in direct contact. However, many humans have strep pyogenes as a part of their normal microbiota. And these individuals serve as a reservoir for the pathogen and can spread it to others. And they themselves can also get sick if they find that the balance of their oral microbiome gets off, or if the bacteria under particularly stressful conditions, they may become pathogenic under certain stressed conditions. Strep throat can be prevented through oral hygiene, keeping your teeth and your throat clean. Because it can ferment sugars, you want to make sure you don't keep a lot of sugars in your mouth. So if you eat sugary foods, make sure you drink a lot of water afterwards, and that will help dilute any sugars that are in your mouth. And if the bacterium can't grow on sugars, then it's hard for it to cause an infection. And avoiding crowds helps with avoiding getting the infection through those respiratory droplets. Amoxicillin is often the first-line drug that is used to treat bacterial pharyngitis or strep throat. But if you are allergic to penicillins or amoxicillin, then something like Z-Pak will also be prescribed. And it is very important to treat the infection. It'll rarely go away on its own. And if it is left untreated, it's more likely to result in those complications of scarlet and rheumatic fever. Bordetella pertussis is our next pathogen. It is a gram-negative bacterium, so that means it has an outer membrane that has lipopolysaccharide or endotoxin. Now, pertussis is going to infect the trachea. And because the trachea is our windpipe, there's a lot of oxygen and air in the trachea. And Bordetella pertussis is adapted to that environment in that it is a strict aerobe. So this is an aerobic infection of the trachea. It is a capsule-forming bacterium, so bacteria do form extracellular polysaccharide capsules. And the cells are these tiny little bacilli, sometimes we call them coxobacilli, because of their rounded yet rod shape to them. Now, the disease that is caused, pertussis or whooping cough, has 2 stages. And when you catch the infection is really critical for treatment. The first stage is when the bacteria are adhering to the trachea cells and the cilia that line the trachea cells. So here's the bacterium, and they actually attach to the cilia that line the trachea. And if you treat the infection in this stage, then the bacteria will die and they haven't produced any toxin yet. So the infection can be treated in the colonization stage, but after colonization and growth occurs, then toxin production begins, and antibiotic treatment will not help the toxin production stage. The infection tends to be mild in adults. You have a thick, heavy cough for a couple of weeks, but the infection will go away. It's much more dangerous in small children and in infants, and the mortality rate is higher in the younger age groups. In addition to capsules and endotoxin, another virulence factor that Bordetella has are fimbriae. There should be an I there. And fimbriae are those extracellular appendages that help bacterium adhere to or attach to surfaces like host tissue surfaces or the cilia that line host tissues. Now, whooping cough is a vaccine-preventable illness. The pertussis vaccine was introduced in the mid to late 1930s. Following the introduction of the vaccine, the number of cases dropped to nearly non-existent. And since the 1980s, and especially since 2000, the number of cases has started to increase again, likely due to the lower rates of vaccinations. There are outbreaks that are reported. That is not uncommon now, and schools are one of the locations where outbreaks are common. And as far as treatment goes, it can be treated in that early stage, in that colonization stage, with an antibiotic. But after the infection reaches the toxin stage, antibiotic treatment will not be effective. Okay, now we're going to talk about the cold, the flu, and then COVID-19. Starting with the cold, the common cold is called rhinitis. Rhino refers to the nose, so rhinitis is an infection or an inflammation of the nose. The symptoms of a cold include a very runny nose at first, followed by a stuffy nose. Sneezing and an itchy throat are also very common. What is not common is a fever. Colds are associated with either no fever or a very low fever, maybe 1 degree, 2 degrees max above your normal body temperature. There are over 200 viruses, different viruses that cause the cold. Not just different mutated strains, but completely different unrelated viruses. Many of them belong to the rhinovirus family. Some of them can be coronaviruses. So coronaviruses are not new to the scene. We do have coronaviruses that can cause mild upper respiratory infections. And then adenoviruses are another family of viruses that can cause upper respiratory cold infections. All right. So the incubation period for the common cold is 2 to 3 days. That means if you start coming down with symptoms, chances are you were exposed to someone who is sick 2 to 3 days before. Transmission occurs by respiratory droplets. So remember, these can come from not just coughing or sneezing, but also through breathing and talking. Smaller droplets will come through our breath and when we talk to individuals. You can also, if you touch a fomite or an object, you can indirectly pick up some of those viruses suspended in some respiratory droplets that have not yet dried. And so the cold can be indirectly transferred through fomites. Because there are so many different viruses that can cause the common cold, there is little immunity against it. So if you have a cold in November, chances are that was caused by one type of virus, and cold viruses circulating in January might be completely different. So once you have a cold during a season, that doesn't protect you from getting colds in subsequent weeks or months, because there are just so many different viruses that can cause rhinitis. For that same reason, there's also no vaccine available. And also there's no vaccine necessary because the symptoms are so mild and the common cold won't result in a life-threatening illness. There are only over-the-counter medications like antihistamines, decongestants, zinc tablets like Zicam can work in reducing the length of the cold. Zinc is known to interfere with viral replication in some of our cells, so this has been shown to have some sort of therapeutic effect in that it can reduce the length of having a cold, especially if you start taking it soon. All right, next is influenza. Influenza is caused by the influenza virus, which is a virus. It has an RNA genome. Remember that inside viruses we can have DNA or RNA, and that DNA or RNA can be single-stranded or double-stranded. So influenza is a single-stranded RNA genome, and it has 2 different types of spikes, and this is where we get some of the names for our different viral strains. So for example, H1N1 comes from one type of H spike, and H stands for hematoagglutinin, and then N stands for neuraminidase. And so the types of spikes that a virus has is what is used to name the different strains. There are 3 general groups of influenza virus strains. The influenza A viruses have both these H and N spikes, and they are often associated with pandemics. Influenza B strains are the common influenza viruses that circulate and cause seasonal flu. Right now, influenza A and influenza B both circulate at sort of the same times in October through February and March. So this year, we had an early peak in influenza B And then later on, most of the cases of flu were caused by an influenza A strain. So sometimes we'll see slightly different symptoms with each one, but the vaccine that is currently available has two antigens for 2 different influenza A strains and 2 different influenza B strains. So when you get a quadrivalent vaccine, that will protect you, or it will induce your immune system to form antibodies against the most commonly reported strains of that flu season. There are influenza C viruses, and these tend to result in very mild flu cases. Most of them go reported because they rarely end up in hospitalizations or doctor visits. All right. And so our regular seasonal flu, or our influenza B strains, undergo changes every year due to antigenic drift or mutations. And this is the reason why we need the flu shot every year, because the spikes change just a little bit on a routine basis. So the RNA genome will mutate just a little bit. Maybe there will be a couple of mutations. And so if the spike or the antigen changes, then the antibodies we had made the previous year might not attach to this new mutated antigen. So the drift is a slow change of the glycoprotein spikes due to a mutation event. Antigenic shift, on the other hand, this is what happens in our influenza A strains. Antigenic shift is when the virus recombines with a second virus inside an intermediate host, like a pig. So if you have an avian flu virus and a human flu virus, both infecting a pig, inside the cells of that pig, if both viruses infect the same cell, there could be a recombination where both genomes kind of mix together, and we have a new virus that comes out containing genes from both the avian flu and the human flu. And so now we have a completely different set of spikes that can cause a severe immune reaction. And so antigenic shift events, this is what will result in the new strains of influenza that are associated with pandemics, like the swine flu pandemic of 2009, and also the 1918 Spanish flu, which killed millions around the world. All right, so influenza symptoms are distinct from a cold. A fever is very common with influenza. A high fever, chills, very strong muscle or body aches, and malaise or just a complete lack of energy. Like the common cold, influenza is transmitted by droplets and aerosols and through direct contact. It is prevented with a yearly vaccination. There are inactivated and attenuated vaccines available. The inactivated vaccines are the ones that come as a shot that you'll get in the muscle of your arm. The attenuated vaccines are the vaccinations that come as a nasal mist. The inactivated vaccines are much more common at pharmacies and doctor's offices. And again, if you have something called a trivalent vaccine, that would have protection against 3 strains of influenza. The quadrivalent vaccines contain the antigens for 4 different strains, typically 2 influenza A strains and 2 different influenza B strains. Because of the rate of mutation, the vaccine is in a good year, up to 70% effective. The effectiveness of the vaccines can be anywhere from 30% to 70% effective. But the more people that get vaccinated, the less likely it is to spread through the community. So herd immunity is really important. The more people that are vaccinated represents more people that will spread the infection, fewer people that will spread the infection to high-risk individuals. And high-risk individuals are more likely to develop complications that could lead to death. And complications will be fluid in the lungs, pneumonia, and these are the types of complications that will result in death. So young children under 2 are at high risk for influenza complications and death. Those that are 65 years and older are also at high risk. Pregnant women and women with, individuals with chronic health conditions and those that are immunosuppressed. Because this is a viral infection, all cases of the flu are caused by a virus, so there's no antibiotic treatment. The only type of treatment you can get is through rest and fluids and letting your immune system just do its job. There are antivirals available which can help lower the length of time that the flu exists, but it's not a cure. It won't make you feel better right away like an antibiotic does with a bacterial infection. But the antiviral will decrease the time that you are experiencing symptoms by interfering with viral replication. So Tamiflu or Valenza are some of the brand names that are associated, and antiviral drugs have to be taken within 72 hours of onset of symptoms. So that means once you experience symptoms, you should see a doctor right away. You can also, there are some pharmacies like Rite Aid that will do flu tests at the pharmacy itself and then hook you up with an antiviral prescription. All right. This year, influenza hit pretty hard. There were 39 million cases of influenza B. We started with a peak in early influenza season this year in October, and influenza B strains dominated those that were causing infections early on in the season before January 1st. Influenza cases that came on in January and February were more likely to be influenza A strains. In the United States, there were over 400,000 hospitalizations and 24,000 deaths. These numbers were part of a range from low to high, so these numbers represent the low end of the range that was given, so they're conservative estimates. Current activity is low. Each week you can check the CDC weekly flu view map to see if it is widespread, sporadic, or if there are regional outbreaks still occurring. Here in Michigan you can see there's still some local activity of influenza. But the flu tests have shown that there are very few positive flu test results right now. And if you're experiencing a severe respiratory tract infection with a cough and a fever, it's much more likely to be COVID-19 than influenza at this time. All right, so that leads us to coronaviruses. We already mentioned coronaviruses when we were talking about the common cold. There are strains of coronaviruses that are already in circulation that can cause mild upper respiratory tract infections. Coronaviruses have a single-stranded RNA genome, and they also have an envelope. That envelope means that they are more susceptible to disinfectants and also very susceptible to hand soap. So the envelopes fall apart if you wash your hands with soapy water. One reason why washing your hands is so effective at getting rid of the virus and preventing the transmission of COVID. All right. There have been novel strains of coronaviruses that have appeared and caused pandemics in recent years. The severe infections have been called severe acute respiratory infections. So there's a sudden onset and the symptoms are very severe. In 2003, we saw the disease SARS, and SARS stands for Severe Acute Respiratory Syndrome. This was caused by a virus, SARS coronavirus, and resulted in 800 deaths. There are very few cases of SARS that have been reported since that outbreak occurred. It's been contained very well. MERS appeared in 2012. This was also caused by a coronavirus that was named MERS-CoV, and MERS stands for Middle East Respiratory Syndrome. This was much more infectious, had a higher deadly rate, and was easily transmittable, but it was contained very early and very well and resulted in about 600 deaths. This year, and starting at the end of the year in 2019, we've had COVID-19, which I know that you have all heard about. This has been caused by a second novel strain of a SARS-CoV virus, so it's related to the virus that causes SARS. When I put up this lecture on Blackboard on April 21st, there had been 37,818 deaths. As of today, there are over 50,000 deaths. So in just a few days, we have gone up by over 10,000 deaths in the United States. All right, so I'm going to show you a video on how coronavirus affects the body, and then we'll talk just a little bit about transmission, prevention, and treatment.
Human, and what should we all do? A virus is really just a hull around genetic material and a few proteins, arguably not even a living thing. It can only make more of itself by entering a living cell. Corona may spread via surfaces, but it's still uncertain how long it can survive on them. Its main way of spreading seems to be droplet infection, when people cough, or if you touch someone who's ill and then your face, say rubbing your eyes or nose. The virus starts its journey here and then hitches a ride as a stowaway deeper into the body. Its destinations are the intestines, the spleen, or the lungs, where it can have the most dramatic effect. Even just a few coronaviruses can cause quite a dramatic situation. The lungs are lined with billions of epithelial cells. These are the border cells of your body, lining your organs and mucosa, waiting to be infected. Corona connects to a specific receptor on its victim's membranes to inject its genetic material. The cell, ignorant of what's happening, executes the new instructions, which are pretty simple: copy and reassemble. It fills up with more and more copies of the original virus until it reaches a critical point and receives one final order: self-destruct. The cell sort of melts away, releasing new corona particles, ready to attack more cells. The number of infected cells grows exponentially. After about 10 days, millions of body cells are infected and billions of viruses swarm the lungs. The virus has not caused too much damage yet, but corona is now going to release a real beast on you, your own immune system. The immune system, while there to protect you, can actually be pretty dangerous to yourself and needs tight regulation. And as immune cells pour into the lungs to fight the virus, corona infects some of them and creates confusion. Cells have neither ears nor eyes. They communicate mostly via tiny information proteins called cytokines. Nearly every important immune reaction is controlled by them. Corona causes infected immune cells to overreact and yell bloody murder. In a sense, it puts the immune system into a fighting frenzy and sends way more soldiers than it should, wasting its resources and causing damage. Two kinds of cells in particular wreak havoc. First, neutrophils, which are great at killing stuff, including ourselves. As they arrive in their thousands, they start pumping out enzymes that destroy as many friends as enemies. The other important type of cells that go into a frenzy are killer T cells, which usually order infected cells to commit controlled suicide. Confused as they are, they start ordering healthy cells to kill themselves too. The more and more immune cells arrive, the more damage they do, and the more healthy lung tissue they kill. This might get so bad that it can cause permanent, irreversible damage that leads to lifelong disabilities. In most cases, the immune system slowly regains control. It kills the infected cells, intercepts the viruses trying to infect new ones, and cleans up the battlefield. Recovery begins. The majority of people infected by corona will get through it with relatively mild symptoms. But many cases become severe or even critical. We don't know the percentage because not all cases have been identified, but it's safe to say that there is a lot more than with the flu. In more severe cases, millions of epithelial cells have died, and with them, the lungs' protective lining is gone. That means that the alveoli, tiny air sacs via which breathing occurs, can be infected by bacteria that aren't usually a big problem. Patients get pneumonia, respiration becomes hard or even fails, and patients need ventilators to survive. The immune system has fought at full capacity for weeks and made millions of antiviral weapons. And as thousands of bacteria rapidly multiply, it is overwhelmed. They enter the blood and overrun the body. If this happens, death is very likely. The coronavirus is often compared to the flu, but actually, it's much more dangerous. While the exact death rate is hard to pin down during an ongoing pandemic, we know for sure that it's much more contagious and spreads faster than the flu. There are two futures for a pandemic like corona: fast and slow. Which future we will see depends on how we all react to it in the early days of the outbreak. A fast pandemic will be horrible and cost many lives. A slow pandemic will not be remembered by the history books. The worst-case scenario for a fast pandemic begins with a very rapid rate of infection because there are no countermeasures in place to slow it down. Why is this so bad? In a fast pandemic, many people get sick at the same time. If the numbers get too large, healthcare systems become unable to handle it. There aren't enough resources like medical staff or equipment like ventilators left to help everybody. People will die untreated. And as more healthcare workers get sick themselves, the capacity of healthcare systems falls even further. If this becomes the case, then horrible decisions will have to be made about who gets to live and who doesn't. The number of deaths rises significantly in such a scenario. To avoid this, the world, that means all of us, needs to do what it can to turn this into a slow pandemic. A pandemic is slowed down by the right responses, especially in the early phase, so that everyone who gets sick can get treatment and there's no crunch point with overwhelmed hospitals. Since we don't have a vaccine for corona, we have to socially engineer our behavior to act like a social vaccine. This simply means two things: not getting infected and not infecting others. Although it sounds trivial, the very best thing you can do is to wash your hands. Soap is actually a powerful tool. The coronavirus is encased in what is basically a layer of fat. Soap breaks that fat apart and leaves it unable to infect you. It also makes your hands slippery, and with the mechanical motions of washing, viruses are ripped away. To do it properly, wash your hands as if you've just cut up some jalapenos and want to put in your contacts next. The next thing is social distancing, which is not a nice experience, but a nice thing to do. This means no hugging, no handshakes. If you can stay at home to protect those who need to be out for society to function, from doctors to cashiers or police officers. You depend on all of them. They all depend on you to not get sick. On a larger level, there are quarantines, which can mean different things, from travel restrictions or actual orders to stay at home. Quarantines are not great to experience and certainly not popular, but they buy us, and especially the researchers working on medication and vaccination, crucial time. So if you are put under quarantine, you should understand why and respect it. None of this is fun, but looking at the big picture, it is a really small price to pay. The question of how pandemics end depends on how they start. If they start fast with a steep slope, they end badly. If they start slow with a not-so-steep slope, they end okayish. And in this day and age, it really is in all of our hands. Literally and figuratively. A huge thanks to the experts who helped us on short notice with this video, especially Our World in Data, the online publication for research and data on the world's largest problems and how to make progress solving them. Check out their site. It also includes a constantly updated page on the corona pandemic.
All right. All right, so as we learned in the video, transmission of coronavirus or COVID-19 can occur through direct contact, through indirect contact with fomites, and also through respiratory droplets. Just like what we talked about with the flu, those respiratory droplets can come from sneezing, coughing, or breathing and talking. The 6 feet for social distancing that you all have heard about comes from how respiratory droplets, how far they travel when you just breathe. So we've got about a 6-foot distance for breathing and talking. And up to 6 meters if coughing or sneezing. So this is one reason why not going to the grocery store, especially if you are feeling any types of symptoms at all, is not a good idea. Because there is no vaccine, our only prevention includes these social distancing measures, staying indoors, keeping space, wearing a mask if you go out into public, as well as disinfection of your household, especially if you're bringing in anything from the outdoors or anything in from another public place like a grocery store, and frequent hand washing. When a vaccine does become available, these control measures will likely, the social distancing measures can reduce, but new control measures until that vaccine is available will likely include massive testing, isolation procedures for those that are sick, and contact tracing. So as there is no proven cure or treatment, there have been some drugs that have shown potential. You all may have heard of the anti-malarial drugs, chloroquine and hydroxychloroquine. These have been used by some doctors as they are already approved for treating other infections like malaria. The results have been mixed on that. So I cannot say whether or not this will be effective or not effective, but there is some potential there. However, looking at an anti-malarial drug is not as effective of an approach compared to looking for an antiviral drug more specifically. So there are some antiviral drugs that have been developed and have been shown to be effective against SARS replication in vitro, which means in a test tube, and they're now being pursued in clinical trials. Clinical trials for both treatments and for vaccines will take time. The minimum time is about 18 months. If you're interested in more resources on coronavirus, I've listed the following websites. We have the CDC has a page for coronavirus. Johns Hopkins University has a page that's keeping track of all of the epidemiology statistics in the United States and worldwide. The World Health Organization is also a reputable source of information, not just for epidemiology and statistics, but if you're also looking for the latest news and research. The state of Michigan has a page dedicated to coronavirus, new press releases, news updates, and then also information about every county in the state of Michigan. And the New York Times has a map that shows all of the counties and their infection rates, the number of infections in every single state, and a total count of infections in the US and the number of deaths. All right, everyone, until next week, take care and have a good weekend.